So first of all, thank you very much, Carl and David, for organizing this seminar sessions, and also for giving me the opportunity to give a talk here. I'm going to talk about the interplay between STDP and synaptic plasticity and rhythmic activity, and specifically we'll focus on the possible contribution of STDP to the transmission of rhythmic activity downstream. I will start with a brief introduction just to try to explain that there is a question, there's something non-trivial which should be considered, and then I will propose that STDP may actually address this problem. We will analyze the issue of transmitting rhythmic activity in two stages. First, we'll look at the most basic toy model of STDP dynamics of a single feed-forward synapse, and then we'll extend the analysis to study entire population of STDP dynamics of a population of synapses in a feed-forward architecture downstream, and their contribution to the transfer of rhythmic activity. And finally, we will get to the part which is still under work, which is the possible contribution of STDP to the distribution of preferred phases in the whisker system. So how can rhythmic activity be transmitted downstream? Okay, so why is there even a question here? So consider a population of an upstream neurons, upstream population of N neurons that are spiking rhythmically, same frequency, presumably, with different preferred phases. So let's assume that the phases are homogeneously distributed on the ring, or even evenly spaced on the circle. Now, if we'll just if the downstream neuron just pulls all the responses of the neurons uniformly, then eventually the entire rhythmic component will be washed out. So we'll just have the DC, the temporal average of the signal. If the downstream neuron will pool the activities of different neurons with assigning random weights, then the DC component will be summed up. Okay, so it will scale like the number of neurons in the population with N, whereas the AC component, okay, because we have interference of different phases, will scale like a square root of N. So essentially, the rhythmic component will be largely attenuated relative to the DC, and eventually we will lose the rhythmic component downstream. So one solution to this problem will be if we can balance, if we have inhibitor contribution that will balance, will cancel out to a leading order the DC component. But this is not the solution that we want to address here. We want to propose a different kind of solution. So consider that instead of having this balanced solution, let's just try to amplify synapses of certain phases and attenuate synapses of other phases. Then in that case, we will be able to transmit downstream the rhythmic component. Okay, and specifically, what we want to focus here is a specific role of activity-dependent plasticity in terms of STDP that may or may not accomplish this task. So you can think of STDP as some sort of an approximation of activity-dependent plasticity that focuses on the temporal relation between pre and post-firing. Okay, so if you think of the results of being poo, then if the temporal relation between the presynaptic firing and the postsynaptic firing is in the causal branch, so x-axis here is the time difference between post minus the pre. So if we are in the causal branch in the results of being poo, then we potentiate the synapse. If we're in the acausal, so if the postsynaptic cell fires before the presynaptic cell, we depress. And over the years, a wide range of STDP rules have been observed. So we have this temporally asymmetric Hebbian learning that being observed in '98, reported in '98, which potentiates the causal branch and depresses the acausal branch. We have something that looks more temporally symmetric, maybe like a difference of Gaussians, and another temporally asymmetric, this time for inhibitory synapse. So we have a wide array of possible temporal kernels for the STDP rule. And what we want to understand is, can STDP actually facilitate the transmission of rhythmic activity by potentiating certain phases and depressing others, and how that may or may not depend on the specific rule, STDP rule that is chosen. So maybe some rules are more suited to the transfer of rhythmic activity than others, and so on. And as I said before, I'm going to address this in two stages. The first stage, we'll consider the simplest toy model you can imagine, an STDP dynamics of a single feed-forward synapse, and we'll assume that you have a presynaptic neuron that is spiking rhythmically, and postsynaptic neuron spiking rhythmically. They are both going to have the same rhythm, but with possibly different phases. So we'll assume that the postsynaptic neuron is probably spiking rhythmically because some other non-plastic input. And we will solve this in the limit of weak coupling. And after that, we want to generalize our discussion to study the STDP dynamics of a population of synapses that arranged in a feed-forward architecture from an upstream population of rhythmic neurons that presumably is supposed to transmit the rhythm downstream to the postsynaptic layer. So we have pre and post synapses, pre and post neurons that spike with a rate that has some sort of a temporal average or DC component of the rate, and some sort of an AC or amplitude modulation component that changes in time with the same frequency for both pre and post, but each neuron may have a different preferred phase. And to derive the STDP dynamics, we write the change in the synaptic weight following Gurion and colleagues and others. We write it as a sum of two terms. First term is potentiation, and the other is depression. And we further assume that we can write the both terms, each of these terms as with a separation of variables. So each term is a product of a function of the synaptic weight dependence and a function of the temporal difference between the post and pre spike times. And of course, we have another latent hero, which is lambda here, the learning rate, which is going to be our hero because it's going to allow us to obtain analytic solution to this these dynamics. It's going to be latent because it's going to happen in the limit of lambda goes to 0. So specifically, again, following Gurion and colleagues, we model the synaptic weight dependence, for example, for the potentiation as a function that decreases the potentiation when we are getting closer to the upper boundary of the synaptic weight. So we want to keep the synaptic weights in the interval of between 0 and 1. And similarly, for the depression, we would decrease the depression when the synaptic weight gets closer to the lower limit, which is 0. We don't want our synapses to change their sign. We have two important variables here. One is mu, that determines how sharp, how the nonlinearity of the synaptic weight dependence. So mu equals 0 is called the additive learning rule, and it's known to be the most multi-stable and the most sensitive to the correlation structure in the upstream population. Okay, this is what we're interested in. Usually, unfortunately, this is hard analytically to address because it has a very it has a discontinuous sharp cutoff at 1 and 0. So it's more convenient to have this continuous variable mu that we can continually get study the limit of mu goes to 0. The other extreme is mu goes to 1. And then in that case, STDP dynamics is more dominated by the dependence on the synaptic weight than on the temporal structure of the pre and post firing, and it tends to be less sensitive to the cross to the correlation structure or the statistics of the presynaptic population, and it also typically also converges to a uniform solution. Okay, so what we want to do is want to derive dynamics for the synaptic weight. So essentially, so if we think of maybe in the line of beam pool results, so a change to the synaptic weight delta W can occur in some narrow time interval delta T if there was a post-synaptic spike in that time interval, and then we have to go back in time, look at for the times where we had presynaptic spikes, compute their delta T, and then compute their contribution to the potentiation term. Alternatively, if you had a presynaptic spike at this time, the current time interval, we need to go back in time, look at the times of postsynaptic spikes, and compute their contribution to the depression term. And since all of our tools come from analysis and differential equations, we want to look at short time differences. So the difference in synaptic weight over short time periods, small delta t. And again, we have the contribution of post spike at this time interval, and then we go back in time, look at the times of the presynaptic firing, and weigh them with the temporal kernel, and so on, and also the contribution for the case of presynaptic spike at the specific time interval, and then we just divide everything by delta t and take the delta t goes to 0 limit, and we get this elegant looking equation for the synaptic weight, which has two terms again, potentiation and depression, with this calligraphic L. However, this calligraphic L, which looks naive, is a random variable, because it is determined by the pre and post-synaptic spike times. However, in the limit of small lambda, essentially W will remain constant over large periods of time on the order of 1 over lambda. Okay, so actually in this case, the dynamics will average the right-hand side over long time periods on the order of 1 over lambda, and in this case, we'll be able to replace the random variable by its mean. So in the limit of slow learning, when lambda goes to 0, we obtain a deterministic dynamics for the mean synaptic weight, and in this case, the distribution is very narrow. That has again a potentiation term and depression term. And each term, it depends on the overlap between the temporal kernel of the STDP rule and the cross correlation between the pre and post synapses. Okay, so in the framework of learning, this sort of dynamics will fall under the category of unsupervised learning. So we don't have a teacher, we don't have a goal to obtain, we don't even have a reward signal that tells us if we're doing better or worse. We just have some sort of a microscopic learning rule that modify the synapses. And so I think that large part of the interest in theoretical neuroscience has been to try and look at what possible computational roles this sort of a microscopic learning rule can yield. Okay, so specifically here we are looking at the ability to transmit a rhythmic activity. Okay, so in this limit, which is termed the mean field Fokker-Planck, learning dynamics, we can show that the again STDP dynamics converge to a fixed point that of course depends on the pre-post correlations, which in the limit of weak coupling, because of the rhythmic activity that we assume for the pre and post neurons, we can fully compute it, and we have a closed expression for the synaptic weight that depends on the various parameters and features of the model. Now, why did I bother you with this extremely simplified toy model? Okay, and it's extremely simplified from several reasons. So one, of course, we're looking at the STDP dynamics of a single synapse, and we do so in the limit of weak coupling. Okay, and two, we're looking at an STDP dynamics which is, in essence, it's by itself an approximation to activity-dependent plasticity, and even as an approximation, we assume this separation of variables, and we had this F plus and F minus that we chose for analytical convenience. So this model clearly is not accurate. And the truth is that no model is going to be accurate. And even if there was an accurate model, we wouldn't be able to analyze it. The utility of this model comes not from choosing the correct 3 or 10 or 7 parameters out of millions of parameters in the biology, but actually from its usefulness. And its usefulness stems from the fact that we have a complete understanding of this model in terms of each and every parameter of how alpha, how the relative strength of depression, how the nonlinearity of the weight dependence, how the temporal structure of the STDP rule and their interplay with the specific rhythm and phase difference affect the resultant synaptic weight. And utility of that for analyzing larger networks would be that this is also what encapsulates our free theory. Because this is what we expand, we build our intuition around, and hopefully helps us understand things, limits that we can't resolve analytically in more complicated scenarios. So in terms of the STDP rule, what makes it useful? It makes it useful that we can understand the role of each and every parameter in this specific parameterization of Guti and colleagues. So what I'm showing you is the synaptic weight profile as a function of the phase difference between the post, which is at 0 phase, and the pre, which is in the x-axis. So the left column is for temporally asymmetric kernel, and the right column is for the difference of Gaussian, so some sort of a Mexican hat kernel. As then you can see, the temporally asymmetric prefers negative phases, so the causal branch, and the temporally symmetric prefer phases that are small in absolute value. When in the additive learning rule, and the limit of mu goes to 0, we have a very sharp transition between the potentiation that goes to 1 and depression that goes to 0. And as we increase mu, we have a smoother synaptic weight profile, but also shallower. Okay, the relative strength of depression you can also understand it intuitively when alpha is less than 1, when potentiation is stronger, then this we have more synapses that will be potentiated, and as we increase the relative strength of depression, the profile becomes narrower, and so on. And you can tease apart and understand the contribution of each and every component in this model. So now we can change gears and study what we wanted. So we assume that we have some sort of a upstream population of neurons that are spiking rhythmically. So you can think of the spiking of each neuron as some sort of an inhomogeneous Poisson process. They all have the same rhythm and same frequency, but their preferred phases are evenly spaced on the circular lattice. Okay, and we can derive the STDP dynamics in a very similar fashion. And again, the dynamics of a synapse of a neuron that has preferred phase phi. So this is the preferred phase of the upstream neuron, and they are all synapsing onto the same downstream neuron. Okay, so phi is the phase of the upstream neuron. Again, we have a contribution of potentiation and contribution of depression, and they depend on the pre-post cross correlation. However, in contrast with the STDP dynamics of a single neuron, where it was legitimate to think of weak coupling, maybe in the limit of large n, here when we allow the plasticity of the entire synaptic population, we cannot assume this limit. So we need to assume some sort of model that will relate the firing in the upstream population to the firing of the postsynaptic neuron. So in order to proceed with our analysis, we assumed, similar to what Gurion et al. Assumed, that a linear Poisson model for the postsynaptic population that is delayed. Okay, so the linear Poisson responds to the spiking of the upstream population weighted by the synapses up to a delay d. Okay, so when we plug this in, the correlation, essentially the cross correlation between the pre and post becomes the cross correlation in the upstream population, which results in sort of an interaction term between the synapses synaptic population. And when we do the math, one finds that the dynamics, the STDP dynamics of a single synapse, and remember we have n such equations, depends on two order parameters, which are W bar, which is the mean synaptic weight of the population, and W tilde, which is the first order component, or actually the population vector of the synaptic weights. Okay, up to some factors that depend also on other factors of the STDP dynamics. And from these equations, we can derive the dynamics for the order parameters, for the mean synaptic weight, and for the population vector. And what we want to study, of course, is the development of phase preference. However, this is not an easy task, because these are high-dimensional nonlinear differential equations, and they are high-dimensional, especially because these F terms, because of the nonlinearity of the synaptic weight dependence, include higher order Fourier modes of the synaptic weight profile. It's nonlinear and it's coupled. So, as I said, our aim was to study the development of a non-trivial phase preference profile. And the problem is that we have a nonlinear coupled high-dimensional dynamics. So the strategy that we'll apply is the same strategy again that Gurion and colleagues applied, and we are going to study exactly what we don't care about. We're going to study the homogeneous solution, and then we'll investigate under what conditions and how does this depend on the STDP rule. Will the homogeneous solution will lose its stability, and hopefully that will signal the development of a phase preference. Okay, so when we study the stability of the homogeneous solution, of course, everything becomes so the homogeneous direction is nonlinear, but it's one direction, and all the others are just can just linearize them around the fixed point, and of course the stability of the homogeneous solution with respect to homogeneous fluctuation is guaranteed because of the nonlinear nature of the synaptic weight dependence that we chose. However, However, instability in the direction of the population vector can arise if the Fourier transform of the STDP rule at the rhythm, or I should have said at the delayed, okay, this is the delay of the post synaptic cell, the delayed STDP rule, if the real part of it is positive. If the real part of it is positive, then for sufficiently low value of mu, we are guaranteed to lose stability of the homogeneous solution in the direction of the population vector. And then, hopefully, phase preference will evolve. And this is what occurs when these conditions are met. And they're relatively easy to satisfy. So what you see here in this plot, you see the 1, 2, 3, 4, four snapshots of the synaptic weights as a function of time. What I'm showing is the average of synaptic weights over 1 minute. So we start with the initial condition where all synaptic weights were equal, and then what you see is this little wiggling of the red line is because of the randomness of the Poisson firing during the first minute, which generates some kind of small fluctuation. However, the STDP dynamics captures these fluctuations and then amplifies them. And already after 30 minutes, okay, what you see here is that the synaptic weight profile, and we arrange the synapses according to their preferred phases of the presynaptic neurons. We see that the emergence of a phase preference, and this phase preference is further amplified and amplified. However, if you look a little bit further in time, well, it seems that the synaptic weight profile does not grow or change its shape. It just shifts. It moves constantly to the left. And you can see this in the synaptic weights themselves. So now here we plot the 120 traces of different synaptic weights, and they all start from the same initial condition, and because of some sort of random fluctuation, some phase is chosen, it's amplified, and then we start seeing some sort of a limit cycle in which synapses are potentiating and depressing and potentiating and depressing. However, you can see that the global order parameters of the first Fourier of the mean and the population vector of the synaptic weights converge to a fixed point, whereas the phase continues to drift in time with a constant drift velocity. So now that we know that the STDP converts to a limit cycle, we can have the limit cycle ansatz and try to solve itself consistently. This is also not very easy because again, because of the nonlinearity of the weight dependence, it involves high order of the Fourier components. But there's a special point here, a pivotal point, which is the special case where the drift velocity, okay, becomes zero. When the drift velocity becomes zero, then essentially all the synapses converge to a fixed point, and the postsynaptic neuron receives a rhythmic component with a fixed DC part and a fixed amplitude and a fixed relative phase. Okay, that doesn't change in time because it's a fixed point. And in that case, this brings us back to our first part of the talk. So we have a postsynaptic neuron that is spiking rhythmically with a constant phase in time, and we have a presynaptic population that spikes rhythmically with specific phases. So in that case, we know exactly how the synaptic weights should look like. Okay, and we are going to use this in a self-consistent manner to understand the mechanism of this limit cycle in the next slide. So what you see here, you see the drift velocity as a function of the rhythm. Okay. Okay, so you see that for certain frequencies, the drift velocity will be negative. For certain frequencies will be positive. But we also have around 29 hertz. We also have a special frequency in which drift velocity vanishes. So let's assume we have vanishing drift velocity. Okay, so let's focus on this graph here. And so if the drift velocity is vanishing, then the postsynaptic neuron is going to spike at a constant phase. So let's, without loss of generality, assume that this is set this phase to be 0. Okay. So if the postsynaptic neuron is spiking in 0 phase, then according to our previous result, we know exactly how the synaptic weight profile should look like for the temporally asymmetric STDP rule, sort of beam pool that potentiates all the negative phases to 1 and all the positive phases to 0. But if this is the profile, then the mean phase, that the input phase of the to the postsynaptic neuron is actually the mean phase of the potentiated region, which is just minus half a pi. So in this case, based on our model, the postsynaptic neuron should fire with a delay d to minus pi, which is a delay of d in the phase. However, for a frequency of 24 hertz, this ends up to be a bit on the negative side. So essentially what will happen for 24 hertz, this will be inconsistent and it will pull the third phase of the postsynaptic neuron to the left, which in turn will pull the synaptic weights profile to the left. Okay, so we will end up with a negative drift velocity. Okay, which will look, and we'll have a synaptic weight profile that will look like this. On the other hand, if we're talking about frequency of 36 hertz, then adding the delay to minus pi will end up with a positive phase, which will pull the postsynaptic neuron to the right, which in turn will pull all of the synaptic weight profile to the right. So we'll have a positive drift velocity. Only in on a special case of 29 hertz, we will have a self-consistent criterion. That's a small note here. For finite population sizes, so if n is sufficiently low, then because of pinning, this region of 0 drift velocity can actually become larger. Okay, so what I hopefully I try to convince you is that STDP can provide a mechanism to shape the synaptic weights in order to enable transition of rhythmic activity. Typically, when that occurs, the weights do not converge to a fixed point, rather they remain dynamic. However, functionality in this case, okay, in terms of the ability to transfer the rhythmic activity downstream, is retained because that we have a stable dynamics for the global order parameters that converge to a fixed point in this case. Okay. Only not all of the global order parameters converge to a fixed point. The phase was arbitrary and drifted in time, but that was fine with us because we did not care about the phase.